Welcome to the deep dive. This is where we take the complex research, the papers, the data, and we break it down for you fast. Today, we're diving into poultry nutrition, specifically focusing on something absolutely critical in this shift to sustainable farming: prebiotics. For a long time, the industry leaned really heavily on antibiotic growth promoters, AGPs. But with things like the EU ban back in 2006, driven by, well, frankly, huge global worries about antimicrobial resistance, AMR producers needed alternatives. And the challenge is massive, right? How do you keep performance, health, feed efficiency up in intensive systems without those AGPs?
It's probably the central nutritional puzzle of our time, honestly. And the most promising sustainable answer involves working with the animal's own biology, its internal ecosystem. Uh, Prebiotics aren't just another additive. They're really a foundational strategy for gut health, both structurally and functionally. So our mission today is pretty straightforward: give you the shortcut to understanding not just what they are, but the really powerful ways they can actually rewire the poultry gut for better performance and safety. Maybe we could start with the definition, because it has evolved.
Good idea. Where did it start?
The original, back in '95, was quite broad, something like non-digestible food ingredients that beneficially affect the host. Pretty general. But the key update came in 2017 from ISCPP. They refined it to a substrate that is selectively utilized by host microorganisms, conferring a health benefit. That word, selectively, is key.
Selectively utilized.
Okay.
So what does that actually exclude now? If the old definition was broader, what kind of things don't quite fit this tighter definition, and why is that selectivity so important?
It's crucial because it demands precision. The aim isn't just throwing indigestible fiber in there. It's about specifically feeding the good guys, the lactobacillus, the bifidobacterium, and essentially starving out the harmful ones. So things that ferment non-specifically, that might accidentally feed pathogens too, they don't really make the cut under this modern definition. That selectivity makes prebiotics a predictable, functional tool, and it helps us separate them clearly from, say, probiotics, which are the live beneficial bacteria themselves, or postbiotics, which are the useful compounds those bacteria produce. Prebiotics are the targeted fuel.
Okay, targeted fuel. Let's unpack that toolbox then. Our sources point to maybe four main types, but maybe it's better to think about what they do. Like some are mainly feeders and others are more like blockers or modulators.
That's a really good way to frame it for practical use. Yeah. The main feeders would be things like fructooligosaccharides, FOS, you get those from chicory, onions, garlic, and also galactoligosaccharides, GOS, which often come from processing lactose, like from dairy. Both of those are excellent at selectively boosting the growth of those beneficial bifidobacteria strains we want.
So they build up the positive side of the gut ecosystem, but then you have these other strategies, the blockers and modulators. The prime example seems to be mannan oligosaccharides, MOS, from yeast cell walls, right? Saccharomyces cerevisiae. The mechanism sounds really interesting.
Oh, it's brilliant. It's a fantastic example of biological intervention. MOS isn't primarily about feeding good bacteria. It's about blocking bad ones. See, pathogens like E. coli and Salmonella often have these little structures, type 1 fimbriae or lectins, that they use to grab onto the gut lining. MOS contains mannose that look very similar to the gut lining receptors. So the pathogens bind to the MOS. Instead, it acts like a decoy.
A decoy, so they latch on to the MOS.
Exactly. They latch on to the MOS, which the bird can't digest. So the whole complex, the MOS with the pathogen stuck to it, just gets flushed right out of the system. It physically prevents colonization.
That physical blocking, that's a really clever non-antibiotic way to reduce pathogen load. Is it pretty reliable? Does it work across different bird ages or pathogen strains?
It's generally very effective against those pathogens that use type 1 Fimbria, which covers a lot of important E. coli and Salmonella types, the ones that are big food safety concerns.
Yeah.
The main thing for efficacy is just ensuring it's consistently present in the gut, so pathogens encounter the decoy before they find a spot on the gut wall. So if MOS is a physical blocker, then the other major player, beta-glucans, work totally differently. They're essentially talking directly to the bird's immune system.
How so?
They come from yeast, fungi, even cereals like oats and barley. And immune cells have specific receptors that recognize these beta-glucans. Binding triggers cytokine production. It's a direct immunomodulatory effect, a real boost to the immune response.
And thinking about cost and where these come from, the sources also mention looking beyond these sort of classic prebiotic sources, right? Exploring things like cereal brands with arabinoxylans or other agricultural byproducts.
Absolutely. That's crucial for sustainability and cost effectiveness. Finding high-value uses for byproducts while still getting these prebiotic effects. That's a major research direction. It ties performance to efficiency and waste reduction.
Okay, so we have feeders, blockers, immune modulators, but how do they actually change the bird's physiology? You said rewire the gut.
Right, this is where it gets really interesting. We're moving beyond just the ingredients. The goal isn't just adding something, it's transforming the gut environment and how the host responds. And the absolute foundation for that transformation is short-chain fatty acids,
Yeah.
SCIAs. When those feeder prebiotics, your FOS, your GOS, are selectively fermented by the good bacteria, the byproduct is SCFAs, mainly acetate, propionate, and the really crucial one, butyrate. Butyrate is, well, it's the rock star. It's the preferred, almost essential energy source for the cells lining the intestine, the enterocytes.
Wait, hang on. So we're not just feeding the bird better, we're directly feeding the cells of the gut wall their ideal fuel.
Precisely. And by powering up these enterocytes, butyrate does amazing things. First, it massively strengthens the intestinal barrier. It physically tightens the tight junctions between cells, these proteins like claudin-1, occludin.
Tighter junctions mean less leakage.
Exactly. Fewer toxins, fewer pathogens slipping through from the gut into the bloodstream. Second, butyrate is a powerful anti-inflammatory. It dampens down inflammatory pathways like the NFE pathway, reducing that low-level energy-draining inflammation in the gut.
So just to put that simply for everyone listening, you're not just giving the bird energy, you're actually reducing the background static of chronic gut inflammation. That has to save the bird a ton of energy, right? Energy it can use for growth instead.
That's exactly it. Wasted energy is wasted performance. And you can physically see the difference. This improved barrier and energy supply translates into better gut structure or morphology. Study after study shows prebiotics significantly increase the height of the villi, those finger-like projections that absorb nutrients, often by 10, even 25%. And at the same time, they decrease the depth of the crypts where new cells are born. This drastically improves the villus to crypt ratio.
More surface area for absorption.
Much more. So the bird can pull more nutrients out of the same amount of feed. That's the core of improved feed efficiency right there.
Okay, a healthier gut structure, better absorption. How does that connect to the bird's overall toughness, its immune system and ability to handle stress?
It links directly. Prebiotics seem to promote something called trained immunity. They gently stimulate toll-like receptors, TLRs, on immune cells. It's like putting the immune system on a low-level alert, not causing inflammation, but making it ready to respond much faster and more effectively when a real challenge like a pathogen shows up. This also seems to boost the humoral response, so you get better antibody production after vaccinations, for instance, against things like IBD, Gumboro disease. And crucially, it helps with resilience against environmental stress. Think about heat stress, massive issue globally. Prebiotics have been shown to lower blood corticosterone, that's a key stress hormone, and also improve the heterophil to lymphocyte ratio, the HL ratio, which is a classic marker for physiological stress in birds. They just handle the stress better.
Wow. Okay, so it's this multi-pronged attack. Feeding the good bugs, blocking the bad ones, physically improving the gut, and making the bird more resilient. What does all that add up to for the farmer, for the producer, and ultimately for us as consumers? This has to mean real, measurable benefits, economically and for safety.
Absolutely. Let's talk numbers because the data is quite compelling.
Yeah, give us some data points.
Okay, for broilers, meat chickens, studies consistently show improvements in feed conversion ratio, FCR. We're seeing anywhere from, say, 1 to 7% improvement.
1 to 7%. That might not sound huge, but in a large operation, that's an enormous cost saving.
It's massive financially. And alongside that, average daily weight gain often increases by 3 to 8%. Some studies with high-dose functional carbs show significant bumps in final body weight, too.
Okay, broilers see gains. What about layers?
Similar story. FOS-type prebiotics, for example, have shown increases in egg production, the laying rate by 5 to maybe 13% in some studies, and importantly, improvements in eggshell quality. Better strength, better thickness, maybe 2 to 4% improvement.
How does that work? How does a gut supplement improve eggshell?
It comes back to those SCFAs, especially the slight lowering of gut pH they cause. This acidic environment actually makes minerals like calcium and phosphorus more available for absorption.
Ah, so better calcium uptake means stronger shells. Makes sense.
Precisely. It's nutrient efficiency translating directly into a better quality end product.
And that enhanced nutrient availability, it's not just good for shells or bones, it links back to that huge public health angle we touched on earlier, pathogen control, right? Prebiotics as a tool for food safety before processing.
Yes, and this is a really big deal. It's pre-harvest intervention without antibiotics. It changes the whole risk calculation. We're talking about concrete numbers here. Challenge studies where they deliberately expose birds show salmonella colonization can be reduced by, say, 25 up to maybe 42%.
42% reduction in salmonella, just from a feed ingredient?
Potentially, yes, in
Yeah.
specific studies. And similar reductions for E. coli, maybe 30–35%. They also help knock down Campylobacter numbers. If you can lower the load of these major foodborne pathogens on the farm before the birds even go to processing, that's a massive win for the entire food chain safety.
That statistic alone, potentially cutting salmonella by nearly half, that fundamentally changes the safety discussion. It underlines why this research is so vital. And we should probably also mention the environmental side effect, too. Lower gut pH, better nutrient use.
Right, it contributes to nitrogen sparing. Better protein utilization means less nitrogen excreted, which means less ammonia in the poultry house, better air quality for the birds and the workers, and less environmental impact.
So multiple benefits stacking up.
Which is why the economic view is shifting. It's less about the cost of the prebiotic and more about its value as, let's call it an insurance policy. The real return isn't just a small bump in growth. It's the increased stability, the reduced risk from disease challenges or heat waves, and also that growing consumer demand for verifiably antibiotic-free meat and eggs.
And producers are getting smarter about how they use them, too. Application methods are evolving. In feed is common, obviously.
Standard, yeah.
But in water application gives flexibility, right? Especially if birds are stressed and maybe not eating as much.
Exactly. Good for targeted dosing during challenging periods.
But the really innovative one, the one that seems like a potential game changer for early life, is in ovo injection, putting it right in the egg. Can you explain that? Why inject prebiotics into the egg before the chick even hatches? What's the big advantage there?
The advantage is getting there first. A chick hatches with a gut that's basically a clean slate, relatively sterile. By injecting a small amount of prebiotic solution into the amniotic fluid, which the embryo swallows just before hatching, we ensure that the very first bacteria to colonize the gut, hopefully those beneficial bifidobacteria and lactobacillus, have their preferred food source available immediately. It's like giving the good bugs a head start. It kickstarts gut development, immune maturation. Studies show it can improve hatchability and lead to stronger, healthier chicks right from day one. It's essentially early life programming for the gut microbiome.
Early life programming, that sounds powerful. But despite all these positive results and clever methods, you did mention earlier that results in the field can sometimes be variable. Why is that? What causes that inconsistency?
Yeah, variability is the challenge we need to overcome for widespread, predictable success. And it often comes down to not quite getting the application right. The specific prebiotic used, the dose, the age and health status of the bird, the quality of the rest of the diet, the farm environment, stress levels, all these factors interact. And crucially, we're often dealing with a nonlinear dose response. It's not always more is better. Meaning if you use too little, a suboptimal dose, you might not shift the gut microbiome enough to see a real benefit. It just doesn't move the needle. But if you use way too much, the excess unfermented prebiotic can actually cause problems itself, like osmotic diarrhea, by drawing water into the gut.
So finding that sweet spot is critical.
Exactly. Precision is key. We need to move away from a generic one-size-fits-all approach and get much better at tailoring the prebiotic type and dose to the specific flock, its age, its challenges, its environment.
And that need for precision really points towards the future, doesn't it? The sources talk about next-generation prebiotics. What are those?
These are really exciting. Think about synthetic oligosaccharides, molecules designed in the lab, specifically engineered to be fermented only by certain highly desirable beneficial bacteria strains. It offers the potential for incredibly targeted modulation of the microbiome, real precision feeding for the gut bacteria.
And even beyond refining the fuel, there's talk of microbiome editing, using tools like CRISPR. That sounds pretty futuristic.
It is, but the research is happening. The idea isn't necessarily editing the bird's genes, but using gene editing tools to precisely manipulate the microbial community itself. Imagine being able to, perhaps, remove specific problematic bacteria or enhance the function of beneficial ones, customizing the gut ecosystem for optimal performance based on the bird's genetics or environment. Still early days, but conceptually powerful.
So to guide all this precision, whether it's dosing current prebiotics better or using these future tools, we need better ways to measure success, right? Beyond just weight gain or FCR.
Absolutely. We need practical, real-time biomarkers, things we can measure easily on farm or in the lab. Researchers are looking at things like the concentration of butyrate in the fecal contents, maybe specific cytokine levels in the blood as markers of immune status, or even taking small samples to measure ileal villus height. We need data points that tell us how the gut is responding so we can make informed, data-driven adjustments to the nutrition strategy in real time.
Okay, so wrapping this deep dive up, it seems clear that prebiotics are way more than just another feed ingredient. They really are this multifaceted, scientifically validated tool enabling a more sustainable, antibiotic-sparing approach to poultry production. They're fundamental modulators of gut health, immunity, nutrient use, kind of the whole package.
They represent an investment in the stability and resilience that modern poultry production absolutely demands, especially in this post-antibiotic era. And maybe that leads us to a final thought for you, the listener, to ponder. Given everything we're learning about next-gen prebiotics, targeted delivery like in ovo, and maybe even microbiome editing down the line, are we moving past just supporting beneficial bacteria? Are we heading towards an era where we can actually, scientifically, program the gut microbiome to perfectly optimize how an animal uses nutrients and handles stress, making its performance almost entirely predictable? The science suggests that future might be closer than we think.